Begin with what is nuclear groove. Nuclear groove appear as sharp, dark, linear line running longitudinally across the long axis of the nucleus. This line is actually an invagination or an inward folding of nuclear membrane. When these grooves are prominent, they bisect the nucleus, creating a highly characteristic of coffee bean. From a pathophysiological standpoint, these grooves are formed due to redundant or excess amount of nuclear envelope, which is relative to overall nuclear volume and forcing the membrane to fold on itself. The groove also associated with papillary thyroid carcinoma, which is PTC, in fine needle aspiration of cytology, FNAC. I will pass this presentation to the next presenter.
Before we go in depth about nuclear groove, let us first look at the normal structure of the nucleus. The nucleus contains several important structures with different functions. First, we have the nuclear envelope, which is continuous with the endoplasmic reticulum. It consists of an outer nuclear membrane and inner nuclear membrane, which are connected at nuclear pores. The nuclear envelope separates the contents of the nucleus from the cytoplasm. Inside the nucleus, we also find the nucleolus, which is the site of ribosomal RNA production and ribosome assembly. It appears as a large, dense structure. The nucleolus forms when chromosomes come together after cell division. Just beneath the inner nuclear membrane is the nuclear lamina, which is mainly made of lamins. It provides structural support, helps maintain nuclear shape, and is important for nuclear integrity and positioning. Nuclear pores, which are embedded in the nuclear envelope, control the movement of molecules such as proteins and RNA in and out of the nucleus. The endoplasmic reticulum, especially the rough endoplasmic reticulum, is studded with ribosome and is involved in protein synthesis. It also plays a role in the movement of proteins and other molecules within the cell. The nucleus also contains chromatin, which is DNA wrapped around histone proteins to form nucleosomes. Chromatin exists in two forms, euchromatin and heterochromatin. Euchromatin is loosely packed and found throughout the nuclear interior, while heterochromatin is tightly condensed and usually located at the nuclear periphery, centromeres, and telomeres. Although centrosomes, microtubules, and intermediate filaments are located outside the nucleus, they can indirectly influence nuclear shape and groove. The centrosome is involved in cell division, microtubules help maintain cell shape and movement, and intermediate filaments provide mechanical strength and help anchor the nucleus in place. Moving on, I would also like to explain a complex that helps maintain nuclear structural integrity, known as the linker of nucleoskeleton and cytoskeleton complex. This complex is made up of cache and sun domain proteins. Cache proteins are located in the outer nuclear membrane, and their cytoplasmic tails extend into the cytoplasm to bind cytoskeletal elements such as actin filaments, microtubules, and motor proteins. Sun proteins are located in the inner nuclear membrane. Their nucleoplasmic domains bind to the nuclear lamina and chromatin, while their sun domains extend into the perinuclear space. The interaction between sun and cache proteins physically anchors the nucleus and allows it to move within the cell.
For this slide, I will explain what nuclear grooves are and their morphological features under the microscope. Nuclear groove is a linear folding of nuclear envelope. Folding is inward. Under the light microscopy, it is a hematoxyphilic line running through the long axis of the nucleus. In order to identify it as a true nuclear groove, the line length must be more than half of that long axis. If less than that, it is not considered as true nuclear groove. Nuclear groove commonly seen as single and longitudinal. Longitudinal refer to the direction of the line. The line must be across that long axis of the nucleus. Multiple nuclear groove can also be seen in a single cell. Next is a morphological feature of nuclear groove under the light microscope. We got 3 nucleus marked with arrow B, arrow C, and also arrow D. Cell marked with B and C arrow show a linear line across the nucleus, which is the nuclear groove. For arrow D, show no linear line at the nucleus, so no nuclear groove present there. Figure 2 is the nuclear groove under the inverted 3D surface plot image. This is used to visualize the contour of the nucleus that will allow a clear visualization of the nucleus. A linear sky blue color can be seen, which indicate the presence of nuclear groove. For figure 3, there is no linear line at the nucleus, so no nuclear groove present here. Although there is sky blue color there, the line is not more than half of the long axis of the nucleus. Lastly, for this slide, it shows what nuclear groove look like under the electron microscope. This is the absolute ultrastructural conformation of the longitudinal nuclear groove cutting through the nucleus. This resembles the coffee bean shape.
The next part, and I'm going to present about morphogenesis, how the groove is actually formed. The groove is not formed in one step, it's formed step by step. In this slide, I'll show the overview of the groove formation. The first one is RET-PTC rearrangement. The second one is cytoplasmic compression with biophysical force. Next, nuclear envelope invagination, which is the nuclear envelope fold inward. Next, chromatin redistribution. And the last one is longitudinal nuclear groove. The first step, I'm going to explain about oncogenic trigger, which is RET-PTC gene. Like I said before, this groove is start with activation RET-PTC gene, which is a cancer cell that actively can order the nucleus to change the shape. This cancer cell give a chemical signal to weaken the nuclear membrane. It actively instructs the nuclear membrane to soften and fold. Okay, you can see in this slide, the normal RET gene change and become rearranged to abnormal RET-PTC gene, which is this can happen within just a few hours, even the cell is not actively dividing. After the activation of the RET-PTC gene, there have cytoplasmic compression and biophysical force that generating inward mechanical pressure on the weakened membrane. For the biophysical force, there is two forces that maintain the nucleus stay perfectly round, which is inward force and outward force. The inward force is generated by microtubule and dynein motor, where the outward force is generated by ectomyosin and vimentin. In normal cell, these two forces in the same value, so it maintains the nucleus in shape. In cancer cell, RET-PTC gene weaken the lamina supporting the outward pulling skeleton. So inward pressure overcome outward resistance. This make the equilibrium break. There are also cytoplasmic organelle around the nucleus that increase the inward force, and this condition make the inward force is become increased than outward force.
So now we move on to the third step, which is nuclear envelope invagination. The term invagination means an inward folding of a structure. In this case, the nuclear envelope folds inward, but it is important to note that the membrane does not rupture or break. It remains completely intact. So what causes this folding? The process begins with activation of the RET-PTC oncogene, which alters signaling pathways within the thyroid follicular cell. And one important consequence is disruption of the nuclear lamina, a protein network mainly composed of lamin AC and lamin B proteins. So the nuclear lamina normally provides mechanical strength and maintains the smooth, rounded shape of the nucleus. When the supporting framework becomes weakened, the nuclear membrane loses its rigidity. At the same time, the cytoplasm exerts continuous mechanical pressure on the weakened nucleus through intracellular organelles and the cytoskeleton. Since the weakened nuclear envelope cannot resist these forces effectively, it begins to fall inward rather than maintaining its original shape. This inward fold generally extends along the long axis of the nucleus. According to the pathological criteria, the invagination usually extends beyond 50% of the nuclear diameter before it is recognized as a true nuclear groove. So next is chromatin redistribution. Normally, chromatin is distributed relatively evenly throughout the nucleus. However, when the nuclear membrane folds inward, the available nuclear space changes. Because of this new shape, the chromatin is mechanically displaced and moves toward both sides of the invagination. Instead of remaining evenly spread, it accumulates on either side of the fold. This redistribution creates the appearance of two nuclear lobes separated by the inward fold. It is important to understand that the chromatin itself is not damaged. Rather, its position changes because the nucleus has changed its shape. Therefore, the groove that we observe microscopically is not simply a line on the nucleus. It reflects a real three-dimensional change in nuclear architecture, where chromatin has been redistributed around an inward fold. Lastly, the formation of the longitudinal nuclear groove, which is one of the hallmark nuclear features of papillary thyroid carcinoma. As the invagination becomes deeper, it creates a prominent longitudinal crease that extends along the length of the nucleus. Under the light microscope, this appears as a thin line or indentation across the nucleus. Because of this deep longitudinal fold, the nucleus resembles a shape of a coffee bean, which is why these nuclei are often described as having a coffee bean appearance. It is important to remember that the groove is not an empty crack or a break in the nucleus. Instead, it is the optical appearance of the intact nuclear envelope folding inward.
Now, I will explain the diagnostic features of nuclear groove in cytopathology. First, what is a nuclear groove? A nuclear groove is a longitude fold or indentation of the nuclear membrane, giving the nucleus a characteristic coffee bean appearance. Nuclear grooves are considered an important cytomorphological features because they help pathologists recognize abnormal cells under the microscope. However, it's important to understand that nuclear grooves are not disease-specific. This means they can be found in several different diseases, so they cannot be used alone to make a diagnosis. Instead, pathologists evaluate nuclear groove together with other nuclear features, such as nuclear enlargement, chromatin pattern, and intranuclear pseudo-inclusions. Looking at all these features together increases the accuracy of the diagnosis. Diseases associated with nuclear groove. Nuclear grooves are most characteristically associated with papillary thyroid carcinoma or PTC. In PTC, nuclear grooves are considered one of the hallmark nuclear features and are usually accompanied by other characteristic findings, including orphan NEI nuclear overlapping, and intranuclear pseudo-inclusion. However, nuclear grooves are not exclusive to PTC. They can also be seen in papillary renal cell carcinoma, chromophobe renal cell carcinoma, and clear cell renal cell carcinoma. Therefore, When a pathologist observes nuclear growth, they do not immediately conclude that the patient has PTC. Instead, they interpret this finding together with the patient's clinical information and the overall microscopic appearance before making a final diagnosis. Compare with clear cell RCC. Another important finding was the higher nuclear growth scores were associated with a higher ferment nuclear grade, which indicates more severe nuclear atypia or a more aggressive tumor appearance. However, the study also showed that nuclear growth alone did not predict patient survival. Instead, high intranuclear inclusion scores were associated with a poorer prognosis in clear cell renal cell carcinoma. Overall, this study demonstrates that nuclear growth are valuable for diagnosis and tumor classification, but they should not be used alone to predict patient outcomes. Next is clinical prevalence. The study found that 69% of the renal cell carcinoma cases show nuclear growth, demonstrating that this feature is very common in RCC. They also reported that nuclear growth were more frequently observed in papillary and chromophobe renal cell carcinoma compared with clear cell RCC. Another important finding was the higher nuclear growth scores were associated with a higher ferment nuclear grade, which indicates more severe nuclear atypia or a more aggressive tumor appearance. However, the study also showed that nuclear growth alone did not predict patient survival. Instead, high intranuclear inclusion scores were associated with a poorer prognosis in clear cell renal cell carcinoma. Overall, this study demonstrates that nuclear growth are valuable for diagnosis and tumor classification, but they should not be used alone to predict patient outcomes.
Okay, to wrap up, let's summarize the key characteristics of nuclear growths and why they matter in a clinical settings. So first, what are we looking at? Morphologically, a nuclear groove is simply a linear folding of the nuclear envelope. On the slide, you will identify these as hematosyphilic or dark staining lines. Crucially, to count as a formal nuclear groove, this line must run along the long axis of the nucleus for more than half of its total length. Next is, why is this important for diagnosis? This groove acts as a key marker for tumor aggressiveness and nuclear atypia. We also see a strong correlation with ferment grading. Essentially, a higher grooves score often points to a higher ferment grade, which signals a more aggressive tumor. So finally, in terms of prevalence, this is a very common finding in renal cell carcinoma, which is RCC. In fact, studies have identified nuclear grooves in as many as 96% of RCC cases. This makes them an extremely valuable feature for pathologists to recognize. So that concludes the summary of our presentation. Thank you.